Hello, I'm Adam Rossano, and I was previously a postdoc in the lab of Dr. Michael Romero in the Department of Physiology and Biomedical Engineering at Mayo Clinic. For this talk, I'm going to be discussing our work characterizing dominant negative effects of a previously uncharacterized point mutation in the sodium bicarbonate cotransporter 1, or NBCE1, as revealed through our use of cellular fluorescent pH imaging and xenopus oocyte electrophysiology. For this talk, I have no relevant disclosures to make, and the competencies covered in this discussion will include medical knowledge and basic science. So our lab primarily studies the sodium bicarbonate cotransporter 1, or NBCE1, encoded by the gene SLC4A4. It is an electrogenic sodium bicarbonate cotransporter found on the basolateral membrane of the renal proximal tubule, where it's responsible for reabsorbing approximately 80% of bicarbonate back from the tubule lumen. Each cycle of this transporter moves 3 bicarbonate ions and 1 sodium ion, which in the charge asymmetry is what bestows its electrogenic nature, so it both generates a membrane potential and is sensitive to membrane potential. 2 copies of the transporter are required to form a functional dimer to get full function of the transport cycle, and the transport is reversible within a physiologic voltage range. The activity of this transporter is vital to both systemic and cellular acid-base regulation, and human mutations in this transporter are known to produce a renal proximal tubular acidosis, glaucoma, cataracts, and band keratopathy. There are previously 12 published human mutations in NBCE1, and you can see them marked here on this topography of the transporter. We are going to be discussing a novel, currently unpublished mutation in one of the intracellular domains. I'll refer to it as either as mutant or G mutant E. This mutation consists of a single point mutation which converts a key lysine into a glutamic acid. As you can see, this takes a very unrestricted side chain and bestows a much larger side chain with a negative charge. The patient that has this mutation presented in childhood with early glaucoma and an elevated intraocular pressure, which required trabeculectomy in early childhood. The patient is also acidotic with a proximal RTA phenotype, and of note, the parents are grossly asymptomatic. So the key questions in assessing this particular mutation was, what are the functional biophysical consequences of the mutation? Are there unique features of the mutation which can provide insight into the structure and function of NBCE1, and are these attributes of the mutation actually observable in human cells? And if so, how would we do such an experiment? So for the initial biophysical characterization, we turned to a xenopus oocyte voltage clamp system in which we injected RNA of the transporter into a xenopus oocyte, and 3 to 5 days later used a voltage clamp setting with a current and voltage electrode to monitor momentary current at set voltages across the membrane. When we do this in a oocyte that's expressing wild type in NBCE1, we can see that the application of bicarbonate and CO2 elicits a large inward current. This is due to the bicarbonate and sodium moving into the cell at this point. However, this current can be reversed with the removal of sodium, indicating that sodium is necessary for the transport activity. And here we see again, with the restoration of sodium in the presence of bicarbonate, the inward current returns and is stopped in the absence of a bicarbonate containing solution. However, in this novel mutation, when we express that in a xenopus oocyte, you can see there is functionally no electrogenic transport activity and no current induced by the application of bicarbonate or the removal of sodium. When we plot current voltage relationships, we can see that for a wild type transporter, you see relatively modest transport in 0 sodium and non-bicarbonate solution. However, in the presence of bicarbonate containing solution, you see a significant increase in transport activity, indicated here by increasing current with more positive voltage. The mutant form of the NBC transporter displays no such increased current in the presence of bicarbonate. To simplify these graphs, we can actually take the linear fit across the voltage range from -20 to +20 millivolts, and use the slope of this, which would be the conductance in microsiemens, to get a more intuitive comparison of these two curves. And when we do that, you can see again the function of this mutated transporter is significantly reduced relative to wild type. So that leads us to the conclusion that this new mutation displays very little transport activity in oocytes. But knowing that MBCE1 functions as a dimer, it begs the question, what would happen if both the wild type and mutant MBC were expressed in the same cell? It turns out that in this case, this novel mutation actually has dominant negative effects, and we can see that here by looking at the conductance of oocytes that have been injected to express either wild type or mutant MBCE1. This is a water-injected control, which has very little conductance. The mutant has significant conductance in the presence of bicarbonate. The wild type has significant conductance in the presence of bicarbonate, but the mutant does not. When we inject twice as much RNA encoding the wild type MBCE1, you can see that there's really no increase or decrease in conductance. If we inject wild type and a previously characterized low function mutation of MBCE1, we can see that that also does not change the overall conductance. However, co-injecting wild type and mutant significantly reduces conductance. This suggests that the mutant form of this transporter is actually able to impair wild type activity when the two are co-expressed. That begs the question, if this can be observed in oocytes, does it actually occur in human cells? To look at MBCE function in human cells on an appropriate scale requires monitoring physiologic parameters that will be tied to MBC function. And to do this, we monitor intracellular pH as MBC moves bicarbonate, which is a base equivalent. To monitor pH in a living cell, we use fluorescent genetically encoded pH indicators. But to achieve this, we actually developed a novel pH indicator that fluoresces in the red range. And you can see the development of this indicator indicated here. So superecliptic fluorine, abbreviated CEF, is a GFP-based genetically encoded pH sensor expressed here in human immortalized trabecular meshwork cells. Fire, our novel indicator, fluoresces red and also expresses well in human cells. This is a version of Fire, the novel red pH sensor, which actually has a slightly increased pKa, making it more sensitive to alkaline environments. And these are three previously published cytosolic genetically encoded pH indicators, which are in the red fluorescent range, known as M-nectarine, M-apple, and Fuji. We see here that when we co-express Fire and superecliptic fluorine in the same cell and expose them to an ammonium pulse, which will induce a predictable set of pH manipulations, both sensors accurately report the same phenomenon, and Fire closely matches the overall dynamic response of superecliptic fluorine, which has been the previous gold standard in the literature. Here you can see calibrations of each of these indicators and how they compare. Superecliptic fluorine has a very large linear range and a pKa around 7.17, indicating its maximal pH sensitivity to be centered at that pH value. Fire has a pKa of 7.4, which makes it near ideal for recording the cytosolic pH of most cells. It also has a linear range and overall response similar to superecliptic fluorine. And you can see here the high pKa variant of Fire, which has a linear range and a more alkaline pH values. M-nectarine and M-apple both suffer from the limitation that they have a very modest overall fluorescence response. It's approximately 1/4 of that observed in Fire. And Fuji, which was previously the best published red genetically encoded pH sensor, has a pKa that's slightly elevated for accurately tracking the pH of the cytosol, and you can see here its linear response is somewhat flat through the predicted cytosolic range of 7 to 7.5. So for these reasons, we believe this newly developed genetically encoded sensor created in our lab is actually probably the most effective red fluorescent pH sensor currently available, and we're hoping to publish this soon. We're able to express this in human immortalized trabecular meshwork cells along with co-transfection of NBCE1 tagged with green fluorescent protein. So this lets us set up an experimental system in which we have some cells which are now overexpressing our NBCE1 and we can monitor pH due to red fluorescence, and other cells which have red fluorescence but no exogenously expressed NBCE1, which would be this cell here. If we expose these cells to a series of acid-base manipulations, in this case, the introduction of a bicarbonate CO2 buffered solution followed by the removal of sodium, we can see predictable pH changes that are indicative of the movement of bicarbonate in and out of the cell. And that's what's shown here. So in our cells that are transfected with the human wild-type NBC, we can see that there is an accelerated rate of bicarbonate influx, indicated here by the recovery of pH during a prolonged CO2 pulse, and that this rate of recovery is very sensitive to sodium. So it is significantly diminished with the withdrawal of sodium, but returns rapidly when sodium is returned to the bath. However, if we transfect these cells with the E91R mutated NBCE1, which is the previously characterized low function variant, we can see that there's effectively no change between the recovery rate and the sodium sensitivity of these pH changes in transfected and untransfected neighboring cells, and those data are quantified here. Looking here at the rate of pH change during the initial recovery phase, the rate of pH change during the removal of sodium, and then resting pH. Resting pH is not different between these groups. However, transfection with wild-type NBC rapidly accelerates bicarbonate loading. Transfection with E91R does not, and the sodium sensitivity is significantly greater in the wild-type transfected cells compared to E91R. So we can use this experimental setup to determine, do the previously established dominant negative effects of this novel NBC mutation occur in human cells, and it turns out they probably do. So here we see the same experiment, only in this case we have an untransfected cell and a cell transfected with the mutated version of NBCE1. What we see here is this bicarbonate loading phase is reduced, the sodium sensitivity is less, and the recovery following the restoration of sodium to the solution is actually less as well. And those data are quantified here. Resting pH is not radically different in non-bicarbonate buffered solutions, but the rate of change is actually diminished compared to untransfected cells, which suggests that the introduction of this mutated NBC is able to suppress endogenous NBC activity in these cells, which would be consistent with a dominant negative effect. So the conclusions of this current work is that we've developed a bright, stable, and responsive redshifted genetically encoded pH sensor known as FIRE, which has spectral properties distinct from GFP. We're currently pursuing whether or not this novel mutation is either simply non-functional or has impaired trafficking to the plasma membrane of the cell, and then following up with further dimerization studies to determine exactly how this novel mutation in NBCE1 is able to achieve the dominant negative effect we've observed. So to explore these options, the lab is currently using chemiluminescence assays to look at surface expression, as well as split YFP constructs to evaluate dimerization between the mutant and wild-type versions of the protein. And we are expanding our methods to evaluate known trafficking and null function mutations to see if the cellular assay can actually be used as a screening tool to predict the nature of defects in either these mutations or any mutations that will be discovered in the future. During my time as a postdoc in Dr. Romero's lab funded on the T32, I also had several other projects ongoing, one of which was to characterize a novel Drosophila oxalate bicarbonate exchanger using pH imaging and Malpighian tubules, as well as fly genetic tools, and then ion selective electrode experiments in oocytes. That's what we can see here. These are Malpighian tubules extracted from a fruit fly expressing a green genetically encoded pH sensor. And we can see here pH responses to bicarbonate, oxalate, and sulfate, with the rate of change being different in a wild-type tubule versus a tubule in which we've used interference RNA to knock down this novel transporter. We've also characterized the ion transport activity of this transporter using pH-sensitive electrodes in xenopus oocytes, and that work is ongoing and should result in a manuscript shortly. Another project was looking at real-time measurement of fluorescent inulin decay in small animals to determine GFR, or glomerular filtration rate. This technology is currently being pursued by Mayo Ventures and hopefully will result in a patent soon, using imaging of fluorescence decay in exposed surface vasculature to actually determine GFR following an inulin bolus. And with that, I will thank all the people I shared the lab with during my 2 years at Mayo, all the people that have contributed to the work, as well as the funding opportunities afforded to me while I was there. And with that, I would take any questions or discussions, except I'm obviously physically not there, so Dr. Romero will be fielding questions in my stead. But thank you for your time and your attention.